This is an Amanita muscaria mushroom. And Amanita muscaria mushrooms, those are your classic red and white, Mario mushroom. Also Alice in Wonderland, right? These are psychoactive. So if you consume these, you're going to have a hallucinogenic psychedelic experience. These were actually used in a lot of like ancient traditions. The Scandinavians would use these. Some of the Vikings would use these before they would go to battle. It's in what's that area in Russia? Siberia. In Siberia, the Siberian shaman. So there was a group of these Siberians, and I don't know how they discovered this, but I think one of them probably lost a bet because he was drinking reindeer's urine. And when drinking reindeer's urine, he realized that you could like have this like crazy supernatural experience. And so these shamans started to worship the reindeer because the urine produced this crazy experience. And then eventually they discovered that it wasn't the reindeer's urine itself, it was what the reindeer were eating. And the reindeer were eating these Amanita muscaria mushrooms. And so they started to herd the reindeer, right? And then they started to actually like worship the mushroom to the point where they started wearing colors that represented the mushroom. They started wearing red and white. And the funny thing about these amanitas is that they generally grow underneath evergreen trees. So in Siberia, Russia, it's like super cold, right? You get evergreen trees all over the place. It's like a perennial type forest. And then in Siberia, it gets very cold. Actually, no, before I get to that part, these are also referred to as fly agarics, meaning that they kill flies. And so these Siberian shamans started to like put the dried mushrooms into their socks, and they would hang the socks around their houses to keep out the flies. And Siberia gets cold, right? So it snows a lot, and sometimes in order for you to get into your house, you have to climb down your chimney. And so, long story short, that's where you get a lot of your Christmas narratives, right? So like the Christmas story, red and white, presents growing underneath an evergreen tree, reindeers, red and white, climbing down the chimney, stockings filled with presents around the house. Yeah, so the Amanita story is really wild. Lots of interesting like history and folklore surrounding amanitas in general. And more importantly for this class, that's where we discovered the muscarinic receptor, Amanita muscaria. And there's like 3 different compounds that are produced by Amanita mushrooms. You get the ibotenic acid, you get mucimol, which is one that we're going to talk about in here, and you also get muscarine. And these compounds generally result, well, first of all, they affect not only muscarinic receptors, but mucimol can also affect GABAergic receptors. And so these can be like sedative type hypnotics. And, that's kind of the effect that they have when you consume them. They can be fatal at high doses. They can cause cardiac arrhythmias. They can cause hyperthermia. So they can actually be pretty dangerous. But generally speaking, this is from an article produced by these guys over here in 2021. The intoxication begins as a state of confusion, dizziness, agitation, ataxia, which means difficulty like walking and doing like tasks, visual and auditory perceptual changes, space distortion, and a lack of awareness of time. So pretty intense stuff. So that's Amanita muscaria mushrooms. We're going to talk about other types of mushrooms next time that are involved in the serotonergic pathway, completely different pathway than amanitas. But like I said, the next couple lectures are going to be very fun. So these are the objectives for today. We're going to talk about the nature and categorization of neurotransmitters. We're going to talk about cholinergic transmission, so acetylcholine, and how it affects muscarinic versus nicotinic receptors. We're going to talk about compounds that contain different types of neurotoxins that affect these neurotransmitters, as well as conditions like neurological conditions that can affect neurotransmission. We're going to discuss glutamate because glutamate is the principal excitatory neurotransmitter, and GABA, which is the principal neuroinhibitory neurotransmitter. And then glycine also has similar effects to GABA. It's going to be another inhibitory type neurotransmitter. So we've already talked about some of these already, but it's going to be a little bit more in depth. So in order for a neurotransmitter to work and be categorized as a neurotransmitter, it needs to be stored in the vesicles in a presynaptic terminal, just like that crappy drawing that I did a few moments ago, right? This is basically what I was trying to draw. So you have the signal coming down from the axon, right? Then you have calcium eventually, well, not in this image. This one's using a different one. This is using sodium and chloride. But pretend that this was a calcium channel. You get calcium coming in, opening up calcium channels, allowing for the vesicles to eventually be released. This diagram is showing a different process than that. But regardless, this idea holds true where you have the presynaptic terminal housing vesicles that contain neurotransmitters. Then the neurotransmitters are going to be released into the synapse, and they have to bind onto a postsynaptic terminal, postsynaptic receptor, and then you get another cascade of signals being sent down to the receiving cells. You also sometimes see co-transmission. So co-transmitters, where you have the release of multiple different neurotransmitters, not just one. Right? Some neurons are going to be more geared towards like GABA, some are more geared towards acetylcholine, but sometimes you can see a combination of neurotransmitters. Now, in terms of categorization of neurotransmitters, the two broad categories are going to be small molecule neurotransmitters. Some examples are going to be your amino acids. Purines can also act as neurotransmitters. Your biogenic amines, right? So you got catecholamines, for example, dopamine, norepinephrine and epinephrine both fit into that category as well. Dopamine is actually a precursor to both of these guys. In the cascade, the biochemical cascade of creating epinephrine and norepinephrine, dopamine is going to be one of the intermediaries for that whole process. Also, your indolamines like serotonin is going to be another one of your small molecule neurotransmitters. Then you have neuropeptides or peptide neurotransmitters. These structures are going to be quite a bit larger. This example over here is enkephalin. Another example might be gastrin. So these are bigger structures, and that's, they fall into the category of neuropeptides. So here are your small molecule neurotransmitters in their broad categories. So you have your acetylcholine, you have amino acids. So amino acids could be things like GABA, glycine. Those are considered amino acids. Then you have purines, and ATP would fall under that category. And then you have your biogenic amines. Catecholamines, for example, fall under that category. And the size of these molecules, you can see the size difference here. They've made these little diagrams that show you the size difference. Your peptide neurotransmitters are way larger, right, versus your smaller molecule transmitters. So I'm debating how much I want you guys to memorize on this list. I highlighted the things in red that I think are very important. So it's good to know your neurotransmitters for this class. So acetylcholine, glutamate, GABA, etc. It's also good to see what kind of effect they have. So remember, glutamate, that is your principal excitatory neurotransmitter. GABA is your principal inhibitory neurotransmitter. You also have some that act both ways. Some act as excitatory, some also act as inhibitory neurotransmitters. And then the way to remove them, because you don't want these neurotransmitters to stay in the synaptic cleft forever, right? You don't want that. You want eventually the neurotransmitter to be either like broken down enzymatically or repackaged, right? You don't want it to linger in the synapse. So there's different things that can come into play to break your neurotransmitters down. So ACHE is acetylcholine esterase inhibitor. Acetylcholine esterase, which breaks down acetylcholine into its respective components. You also have transporters that basically transport the neurotransmitter back into the presynaptic terminal. Neuropeptides are going to be broken down by proteases, right? Proteases break down proteins, right? So there's different ways that you might get breakdown of these neurotransmitters. MAO, do you guys know what that stands for? That's monoamine oxidase. And so monoamine oxidase is going to break down, for example, serotonin. It's going to break down dopamine. It's going to break down norepinephrine and things like that. If you took a monoamine oxidase inhibitor, that would prevent the breakdown of those neurotransmitters. So there's certain like antidepressant drugs. That's how they work. They basically inhibit the breakdown of the neurotransmitter. So you get serotonin lingering longer within the synapse. And then you have reuptake inhibitors like selective serotonin reuptake inhibitors, SSRIs. They prevent the reuptake of serotonin in the synapse. And so different drugs have different mechanisms of action, and they usually affect one of these types of pathways. They either affect the the receptor site directly, or they prevent the breakdown of the drug, right? Remember with like neostigmine or physostigmine or edrophonium. Remember that tinsilin test with myasthenia gravis? Remember that's how they were like diagnosing myasthenia gravis. They administered the edrophonium. It's an acetylcholinesterase inhibitor, right? Then it allows acetylcholine to stay in the synapse for longer. And that's why in that video that I was showing you with that kid, right, all of a sudden he was able to move his eyes again, open his eyes properly. So different drugs will have different mechanisms of action, and it could involve these types of mechanisms. Again, I don't know if I don't really care about the whole like cascade of producing these. So we're going to talk about some of the cascades because they are kind of interesting, right? For example, like dopamine being a precursor for epinephrine, norepinephrine, like that's really interesting. Glutamate is also a precursor for GABA, which is kind of ironic considering that glutamate is excitatory, GABA is inhibitory, right? But it's all part of the biochemical process. So, but I'm not going to test you guys on the precursors because that would be really mean. I'm also definitely not going to test you on rate limiting steps in the synthesis of these. Again, that's like super, super mean. I don't care about the size or the types of vesicle, but mechanism of removal, the effects of the neurotransmitter and the neurotransmitters themselves, I think that's going to be fair game for your guys' next exam. I'm thinking all of them, most of them at least the ones that we talk about. For sure, acetylcholine, for sure, glutamate, GABA, glycine, catecholamines, serotonin, histamine, maybe not so much ATP, maybe not so much neuropeptides, definitely endocannabinoids, and definitely nitric oxide. But I don't know about the other ones. I'm still going to debate on that. So I'll let you know, though, before the exam on which ones to specifically focus on. Acetylcholine, that was the first neurotransmitter discovered. So this is like the OG neurotransmitter. And it's going to be made up of acetyl-CoA and choline. Where have you guys heard of acetyl-CoA before? Yeah. More specifically, glycolysis before you get to the citric acid cycle, right? When you have citric acid cycle in the process in the presence of oxygen, then you get pyruvate eventually turning into acetyl-CoA, getting funneled into the citric acid cycle. So acetyl-CoA is the first step in that whole process. So this is going to be one of the products, right? Acetylcholine comes from that whole process. And then you also have choline, which binds combined together. That's where you get acetylcholine. And it's very efficient. It's very effective at doing what it does. You need to have different compounds to be able to produce acetylcholine. So you have to have, example, choline acetyltransferase. You have to have pyruvate to create acetyl-CoA. And yeah. So again, I'm not going to test you guys on the biochemical pathways of this. I'm not going to test you on how to produce these neurotransmitters, but just know what the overall effect is going to be. Now, acetylcholine can bind to either nicotinic or muscarinic receptors. Nicotinic receptors, why are they named nicotinic receptors? Is because they bind to nicotine, right? So nicotine is one of the things that could bind to these. Also acetylcholine as well. Here's a dude. He broke the record back in 1995 of the most number of cigarettes smoked at one time. 800 filtered cigarettes smoked. I forget how long it took. It took him not very long. It was like 8 minutes or something like that. And he felt pretty crummy afterwards. And I didn't put the full quote that he had. He was like, yeah, I feel kind of crummy now, but I'm going to get over it. It's like, wow, dude. Pretty hilarious. Anyway, so nicotinic acetylcholine receptors, when they get stimulated at the neuromuscular junction, they're gonna act as an ion channel. And so when they get stimulated, they allow for cations to move through. It's not just sodium. It could also be potassium. It could also be calcium as well. And then again, acetylcholine is going to act as a ligand, but other things can act as ligands on these receptors as well, including toxins, right? So Bungora toxin, which comes from a type of snake. It's going to be a Taiwanese snake that can bind to this receptor, causing some issues. But the fact that you can use toxins to bind to these receptors means that you can use these different types of toxins to study how these receptors actually operate and function. Another thing that I don't really care about too much for this class is going to be the actual structure of these receptors. So not super important. The structures are going to have like similarities across the board. Like if you're talking about an ion type chemical receptor that opens up an ion channel, they're all going to kind of have these different types of subunits. They have sometimes transmembrane proteins that go through the entire membrane. And then at the end of the day, long story short, once they get stimulated by the neurotransmitter, they open up or they close, right? So that's kind of the fundamentals to what these ion channels do. So you have different subunits. Some of these subunits are going to be responsible for the actual binding of acetylcholine. Like for this one, it's the alpha subunit. And when they bind to the neurotransmitter, they change shape or they change their conformation. Right? That's the long story short here.
Can you explain the nicotinic side?
Sure. What do you want?
Just breeze over it.
So it's called a nicotinic receptor because that's how we found out that we found out how it functions, right? It was discovered that nicotine bound to it. But in reality, in terms of physiology, it's acetylcholine that's going to be the one binding to it, right? But it's called a nicotinic receptor because it binds to nicotine, right? But physiologically speaking, this is how your muscles move, right? Nicotinic receptors are found at the motor end plate of your neuromuscular junction. So if you were to smoke a cigarette, right, or to have like any sort of like tobacco products, they're going to be binding to these receptors. And sometimes receptors and things like that are named after how they discovered, like what toxin. Muscarinic receptors binds to muscarine from the Amina muscaria mushroom. That's how they found out how that works.
So that's why it causes like a relaxation?
Yeah. Nicotine is interesting. It can cause relaxation and it can also cause stimulation as well. In certain regards, it might be dose dependent. Like low dose might cause more of a relaxation. High dose might cause more stimulation, or it's the opposite. I can't remember off the top of my head, but there's, some of these are going to be dose dependent. These receptors have many, 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 many subtypes, and I am not going to hold you responsible for all the subtypes. So for example, serotonin. This is not even like the full list of subtypes for serotonin receptors. Serotonin receptors have tons of subtypes. So like, for example, in my research project, the receptor types that we're looking at for one of the compounds that we're using is the 5-HT1A and 2A receptor. So those are subtypes of receptors. They're not even listed on this list here, right? There's tons of them. There's tons of acetylcholine subtypes, AMPA subtypes, NMDA subtypes, GABA subtypes, right? Look, alpha 1 through 6. The subtypes have subtypes. So there's a lot of subtypes, and they all do different things.
What's like an example of why there's subtype?
Because sometimes they might have different effects physiologically. How can I think? Let's keep it simple so you guys have a good point of reference. Think about adrenergic receptors, right? You got alpha, you got beta, right? You got alpha 1, alpha 2, you got beta 1, beta 2, right? Those are all subtypes of an adrenergic receptor, right? So catecholamines like epinephrine or norepinephrine, when they bind to alpha 1, it causes stimulation. Bind to alpha 2, it causes inhibition, right? And they're going to be found in different parts of your body. Alpha 1, you find it in your blood vessels, right? Beta 1, you find in your heart. Beta 2, you find in your lungs, right? So these are receptors that you find in different areas in your body, and whenever they get stimulated, they do various different things.
Even though they're using the same.
They're using the same neurotransmitter, but the receptor subtypes are going to result in a different effect, right? Does that make sense?
Yep.
And quite honestly, I don't even know what a lot of these subtypes do. There's a lot of them. It's interesting. I mean, super interesting. And again, different types of compounds can have different effects on these types of receptors. So these are the ones that are affecting neurotransmitter receptors. So they can either be excitatory or stimulatory type synapses. So you got that Bengora toxin, which I mentioned, that comes from a snake. That's going to block nicotinic receptors. So what does that mean? If it irreversibly blocks a nicotinic receptor, what does that do to the person who gets exposed to this toxin? Paralysis. Yeah, absolutely. So that's how these snakes work. They bite their prey, it paralyzes their prey, and then they eat their prey. right? That's how these toxins are used by different animals. Alpha conotoxin, which comes from a cone snail. They shoot this stuff out to kill prey as well. They kill little tiny fish with these things. And again, it blocks the neuromuscular junction. Aracholine, I think I'm pronouncing that correctly. It's an agonist to acetylcholine receptors, so it would overstimulate those receptors, which could also cause paralysis. That would cause like spastic paralysis, right? Scopolamine, that blocks muscarinic receptors. What happens if you block a muscarinic receptor in your heart? Huh? It would make your heart pump more, right? So, yeah, so atropine, this is a drug that's used for patients that have heart failure. And where does atropine come from? It actually comes from Atropa belladonna. What does belladonna mean? It means beautiful woman. Why is it called that? Because back in the day, women used to actually take this stuff and drop it into their eyes, and it would cause dilation of their pupils. And, you know, us guys are suckers for a gal with dilated pupils. And so belladonna, it makes them beautiful. Why is that the case? Because usually when someone sees something that they like, their pupils dilate, right? You can especially see that with a person with blue eyes, right? You can really see their pupils dilate. So if you see someone look at you and all of a sudden their pupils dilate, they're like, oh, they like me, you know? And so that's why women used to do that back in the day. So belladonna, they turned it into a drug, and that's where you get atropine, used for heart failure. So if a patient has heart failure, this drug blocks the muscarinic receptor, so blocks acetylcholine from binding to it. And what happens? You get, instead, the sympathetic nervous system takes over, right? So you get sympathetic activation of the heart taking over, right? No more parasympathetic activation. So in other words, your heart can increase contractility, and so it can continue to function properly. Alpha agatoxin blocks glutamate receptors, right? Remember, glutamate is going to be one of your principal excitatory neurotransmitters. So different types of compounds causing different types of effects. Right? So here's your banded crate snake. Pretty cool looking. Gross. You're not a reptile fan? I like snakes. I think they're pretty cool. They're creepy though, that's for sure. Here's your venomous dart. The cone snail that shoots a little venomous dart. That's how it kills its prey. So super, super cool. Beetle nuts, or betel nuts, I think that's how you pronounce that. Another type of plant that causes toxicity. Here are some other things. So here are what happens when you block inhibitory synapses. So strychnine. So one of the mechanisms of strychnine is going to be that it blocks glycinergic receptors. Remember, glycine and GABA are going to be like two of your major inhibitory neurotransmitters. So if you block glycine, that's going to cause overactivity, right? So if you block GABA, that also causes overactivity, right? Because these two slow the brain down, right? Drugs that are called depressants, right? Like alcohol, for example. When you drink alcohol, it's a depressant. It doesn't mean that you're like, crawling in the corner and you're crying, right? Although that could happen, right? But it's a depressant because it slows down the neuronal firing of your brain. Because one of the things alcohol does, it acts on GABA receptors. So if you block those, inhibitory receptors, you're going to cause excessive stimulation. So that's why if you use strychnine, you're going to see seizures, excessive neuronal stimulation. If you block GABA receptors, again, you also get excessive stimulation. So here's Anamarita cocculus, picrotoxin that blocks GABA receptors. Insecticides can also block GABA receptors. And here's mucimol. Mucimol does the opposite. It doesn't block the GABA receptors. It induces GABA receptors. So it causes kind of like a depressive type of effect. It's an intoxicant. Like people used to use this stuff, and they still do recreationally, right? It gives you kind of like a drunken feel at low doses. At higher doses, it can make you start hallucinating. Muscarinic receptors also binds to muscarine receptors, so it can cause a parasympathetic state. So if you take a lot of these amanita mushrooms, not only might you get like the sedative effect from GABA, but from the muscarinic effect, you also go into a parasympathetic state, which is why it could be very dangerous to the heart, right? If you take too much of it, it can make your heart go really, really slow because you're inducing an excessively parasympathetic state. So we already talked about myasthenia gravis, but let's just kind of briefly go over it again. So myasthenia gravis, again, autoimmune disease. It breaks down the nicotinic receptors at the motor end plate. And the way that you treat that, again, is with acetylcholine esterase inhibitors, just like neostigmine. That's, edrophonium does the same type of thing, right? That's how you can diagnose it. Here is the neurons firing on an individual with myasthenia gravis. A normal person, you can see normal neuronal firing, and it's sustained, right? Versus when you have myasthenia gravis, once you keep on stimulating that muscle, right, you're not going to get a sustained contraction over time. So that signal starts to get reduced. That's why with myasthenia gravis, patients will get tired with repeated use of those muscles. And then if you treat the patient with something like neostigmine or edrophonium or anything like that, that prevents the breakdown of acetylcholine, then you see a little bit of a regaining of the function. It's not perfect though, right? It's like the nicotinic receptors are still being destroyed. They're still being destroyed. What you're trying to do is you're trying to increase the amount of acetylcholine in the synaptic cleft. It's not going to cure it, but it can at least alleviate some of the more severe symptoms of myasthenia gravis. Yes?
I forgot to ask, do you want us to know like all the different kinds of like neurotoxins? The derivations?
No. Mucimol is a really good one. Strychnine might be a good one to remember. I'm not going to test you on these other ones over here.
What about the ones for the neurotoxins that act on neurotransmitter receptors?
Let me think here. Atropine, I will definitely include that. Atropine, especially because it's a commonly used pharmacological drug, right? Lots of people take it for heart failure. I might have to come up with a little list of things for you guys to focus on, because I don't want you to learn all of these, but I definitely want you to know a handful of them. So let me chew on that. I'll put together like a list for you guys, and then because you guys, your next exam is going to be after spring break, and so I'm going to be writing your exam probably during spring break. Thursday. Yeah, this will be. Yeah, we can do like a little review, or we could do a lecture or whatever. Maybe a review session would probably be a good idea. So, good question. I'll have to show it. Let me actually make a little note here just so I remember to put that list together. So, okay. Cool. All right, so here's muscarinic receptors. So remember, nicotinic receptors, that's going to be especially important for muscle contraction, right? It's an ion type channel, right? When you stimulate it, it's going to be an ion channel. It opens up cations. It opens up for cations, sodium, calcium, and potassium. The muscarinic receptor is a little bit different. This one's going to be a G protein coupled receptor. It's metabotropic. Now, because of that, it's going to be slower, right? Remember, the ionotropic receptors are much more rapid. Metabotropic are going to be a little bit slower. They can be both inhibitory or excitatory. If it's inhibitory, they're going to open up potassium channels, right? Potassium channels are going to allow for potassium to move in which direction? Inside or outside of the cell? Outside, yeah. It's going to move outside, bringing the charge of the cell hyperpolarized, making the cell less prone to firing. So that'd be the inhibitory effect. If it's excitatory, then it's going to close potassium channels, right? And so it's going to allow for more of it's going to allow for more like depolarization type of effect. And these types of muscarinic receptors are going to be really important for the autonomic nervous system, right? The autonomic nervous system, that's going to be especially mediated by things like the vagus nerve, right? Cranial nerve number 10. This is going to be the mechanism of these, right? So the autonomic nervous system, vagus nerve stimulation of the heart is going to release acetylcholine at the heart binding to muscarinic receptors, slows the heart down, right? That's why if you take a drug like atropine, you block that muscarinic receptor, you're not going to get that effect. So you're only going to get sympathetic stimulation. Smooth muscle contraction, right? Smooth muscle contraction, that's autonomic, involuntary. That's going to be involving acetylcholine on muscarinic receptors. Exocrine glands can also be stimulated by this process. Okay, so lots of different parts of the body, mostly autonomic, is going to be involving muscarinic type receptors. Again, G protein coupled receptors, that's why they act a little bit slower, right? Nicotinic receptors, think about your muscle contraction. How fast is that? It's instantaneous, basically, right? Versus like a smooth muscle contraction, it's much slower. Have you ever seen videos of peristalsis? You can watch videos of peristalsis, and it's very slow, comparatively speaking, right? So that's going to be involving muscarinic receptors. Same neurotransmitter, but different type of receptor. Again, you are not responsible for all of these different subcategories of receptors. Oh, here they actually list those 5-HT1A, 5-HT2A receptors. Those are the ones that we're looking at for my lab. But there's tons of them. You're not responsible for all of those. So let's now talk about glutamate. Again, glutamate is the principal excitatory neurotransmitter. The short hand is going to be GLU or GLU. It's going to be synthesized from glutamine. And here's the pathway. So you get glutamine, you have glutamate synthase, which is the enzyme that now allows for the production of glutamate. That can also be turned back into glutamine using glutamine synthetase, rather. I am not a biochemist by any stretch of the imagination, so these types of enzymes and stuff kind of like make my head spin. Some of my colleagues would probably be much more into this than I am. But here's the thing that I do find very interesting. The fact that GABA is derived from glutamate. And again, remember, glutamate, main excitatory neurotransmitter. GABA, one of the main inhibitory neurotransmitters, but they're relatives to each other, right? In the presence of vitamin B6, that's one of the cofactors that allow for the production of GABA. So super cool stuff. Again, I'm not going to test you on those pathways, but I do find them pretty interesting. So here's what's going on with glutamate. Glutamate gets released into the synapse, but again, you don't want that to stay in the synapse forever. You want it to eventually clear out. And so part of it being cleared out, it gets sent into a supporting glial cell. Remember, your glial cells are supporting cells. So it gets sent into a glial cell. It can also be sent into the actual presynaptic terminal. So it goes right back in to the presynaptic terminal, or it could go into the glial cell. Then it goes through a whole process where it gets turned back into glutamine, and then it goes back into the presynaptic axon, eventually going back into the process of turning it into glutamate, where it can thereby be put into a little vesicle so it can be prepared to be released again. What's going on here? What does this whole process mean? Like, big picture. You guys like to recycle? Right? I think recycling is a waste of time. They don't actually even like do anything. I've heard so many stories about like the stuff that they put into recycling actually just goes into the dump.
That's what I heard too.
Yeah. So it just makes people feel better for the most part. It's like, oh, I'm recycling. But in reality, it's like, are you really? So the body likes to recycle things, right? This is for efficiency, right? You have these things, the substrates that are already in place, right? You can create different compounds from different substrates, right? Think about like acetyl-CoA, right? That's not just going to be used for the citric acid cycle. It could also be used to make acetylcholine, right? So there's a lot of like promiscuity with these different molecules. And with, this, you're recycling the neurotransmitter, right? That's a good thing. So you can reuse it for a later time. So that's the big picture story on neurotransmitter recycling. For glutamate, there's going to be a couple different receptors. One of the most common ones that you're going to find is going to be the NMDA receptor. You're also going to find AMPA receptors or AMPA. And then you also have these kainate receptors as well. So the AMPA receptor, this one is going to be really, really rapid. So it's super fast, and it's going to be an excitatory postsynaptic current type activation, right? So it's going to be excitatory. NMDA receptors are going to be, comparatively speaking, a little bit slower. These are going to allow for ions to come in, so calcium, sodium, potassium, as well as magnesium. It could cause hyperpolarization if you block magnesium, or it could cause depolarization if you allow magnesium to be released from the cell. In other words, you have like kind of a balancing act. And the idea behind that is that these are like little tiny mechanisms that allow for basically information to get stored. That's kind of the idea behind this. So glutamate is going to be really important for information storage. So whenever you have glutamate activation in your brain, that could be part of like learning and things like that. Kainate, we're not going to really talk about this one so much, but it does rise pretty quick, but it also it decays a little bit slower. So here's a couple graphs that show the effects of these different receptors activation. So AMPA, again, AMPA is going to be really, really rapid, and then it decays rapidly as well. NMDA, it's going to be a little bit slower, and then you get kind of like a prolonged decay. And kainate, it's going to have a little blip, so it's going to be a little rapid. It's not going to be as intense as the AMPA activation, but then it's also going to decay pretty rapidly. And then you find these in different areas. So like this study over here was looking at these receptors in the visual cortex, right? You had interneurons that are found in the visual cortex that are using these types of receptors. You can also see these in areas like the hippocampus, right? So in the hippocampus, again, like if you're talking about information storage, hippocampus, that's where you store a lot of your, long-term memories, right? So you're getting information storage mediated by these types of receptors. So super important stuff. Again, I don't really care so much for this class, the structure of the actual receptors, but these are what those AMPA and kainate receptors look like. They've got those little like clam-shaped, it looks more like Pac-Man to me, but the clam-shaped binding site is where you have, for example, glutamate binding and then actually turning on or actually opening up the channels. And so you have those transmembrane proteins. Transmembrane proteins open up when you have binding to those neurotransmitters. But like I said, not super high yield for this class. Here's another mechanism of what's going on with these receptors. So you have your NMDA receptor, which is going to be voltage dependent. And so when it's not being stimulated, then you're going to have magnesium binding inside the channel. So it basically blocks the channel off. Then once you have depolarization, then magnesium basically gets funneled off, and then it opens up the ion channels for other ions to come rushing in. Sodium, calcium, etc. Yeah.
So, a lot of my friends take depression medication and they can't drink alcohol. Is it because it causes, it counteracts it? I feel like someone asked this question to you.
Yeah, someone did ask a similar question. It's like if you're on a certain medication, like anti-epileptic drugs, for example, you probably shouldn't drink alcohol. Yeah, so like alcohol does act as a depressant, and you could get like a multiplying effect. Or if you're taking one type of medication and you couple it with another medication or another drug, then you get like an amplifying effect, and it could potentially be dangerous. I don't know what type of antidepressant your friend is on, but I'm assuming it's an SSRI. SSRIs, I mean, in terms of alcohol, I imagine that it's... Does she really? So it makes it worse? Yeah. I mean, alcohol can mess with your brain chemistry. So that's all I can say about that.
It just has a different effect on
Everyone's biochemistry is going to be different. Like what affects me might affect you completely differently, right? So that goes across the board with different drugs. I mean, that's why they have different types of like SSRIs, right? Maybe Zoloft works for someone better than Prozac, right? So some drugs work for other people. Like we're learning about anti-epileptics, right? In patho, there's tons of different anti-epileptics. Carbamazepine, there's Keppra, there's Depakote, there's lamotrigine, Lamictal. There's tons of different meds.
I try and explain to my business major friends that everyone's brain chemistry is different. So the reason why I don't want to drink alcohol is because I don't feel the same. And they're like, that's not this thing. And I'm like, it is this thing.
That's why they're business majors. That is why they're business majors. No offense. It's important. We need our business majors. And we also need our communication majors. You're a business minor. That's okay. We'll forgive you. That's so funny. So then going back to these receptors over here. So these are this is a line chart that's showing the effects of removing magnesium from the mix, right? So if you remove magnesium, that's going to affect the glutamate channels in different ways, right? So you get no blockage of the channel pore. In other words, calcium, sodium, all those different ions are going to be able to enter into the cell regardless, right? So you don't even need to have glutamate stimulation to allow that to happen. Again, these are the structures of these different receptors. They got those little clamshell receptor sites or binding sites. Once they get bound, then they change conformation, allowing different downstream activation to take place. So here's another type of domain. They call this one a Venus flytrap domain. Do I see a Venus flytrap? I don't, but apparently you have to use your imagination for these sorts of things. But that's where the glutamate receptor is. So that's where you're getting binding to the glutamate receptor. And this one in particular is going to be a G protein coupled receptor. So this one is going to be slow because it has to allow for a downstream cascade to take place. You get changing of the conformation of the G protein. And once you get that taking place, then you get the stimulation of the effect of the receptor. So now let's talk about GABA. What does GABA stand for? It's going to be gamma aminobutyric acid, but for the sake of this class, we're going to refer to it henceforth as GABA, because that's a mouthful right there. What type of drugs and compounds affect GABA? There are a lot of drugs that affect GABA. First and foremost, alcohol. That's one of the main mechanisms of alcohol. But alcohol is a very dirty drug. It actually affects lots of receptors. It affects dopaminergic receptors. It affects serotonin receptors. It affects opioid mu receptors, right? So alcohol does lots of things. Alcohol can stimulate, right, through like dopaminergic activation. That's another reason why it's addictive too, is because you get dopamine surge, right? Dopamine mediates addiction and just repetitive type behaviors. Serotonin make you feel good. Opioid receptor make you feel good. And then you get GABA, which is part of the whole reason why alcohol is considered a depressant, right? It's going to slow the overall stimulation of the brain down.
Does it affect your prefrontal cortex?
It affects your whole brain. Absolutely. Why do you think when people at the Phoenix Open were taking their shirts off and sliding down disgusting grassy hills? It's because they're drunk and affect their prefrontal cortex. Maybe if they're sober, they wouldn't do something like that.
Also, like, why is it that people, like, if they're thinking of, like, not doing...
Oh, I know what you're saying, because maybe you're blocking the frontal lobe that would tell you.
Yeah,
Shouldn't-
inhibitions. Yeah,
Insane.
that's the reason why it reduces inhibitions.
You, like, think of something, or you're like, you know,
You probably shouldn't do that.
or you shouldn't say it,
Yeah.
and it all comes out when you're...
Oh, yeah, it all comes out when you're drunk. Yeah, absolutely. That's why it's a social lubricant. What else does that too? Barbiturates. Truth serums, quote unquote, they're usually barbiturates, and they're hypnotic sedatives. Some barbiturates have been used in hypnosis. They'll actually like give the patient or the person that's undergoing hypnosis, they'll give them barbiturates, and it will like basically remove all their inhibitions. And so they'll actually start talking about things that, you know, are in their mind, right? Things that they probably wouldn't otherwise think about, right?
I wonder if therapists do that.
They do. They have in the past. They've done this like with like hypnosis.
Really deep.
Yeah, absolutely. Yeah. So you'd have to be a psychiatrist to do it, because only psychiatrists can administer actual meds. If you were a psychotherapist, you would have to be working in conjunction with a psychiatrist to be able to administer those types of drugs. But barbiturates can be used for a lot of things. They've also been used in the past to treat depression. They've been used also to treat epilepsy. The problem with barbiturates is that it's really easy to overdose and die on barbiturates. And so that's why psychiatrists are a little hesitant to be prescribing barbiturates. Benzos, on the other hand, benzodiazepines can be addictive, right? But they're a little bit safer than barbiturates, so they're good at treating things like anxiety, right? They're anxiolytic drugs, right? Lots of people take lorazepam, lots of people take, like Xanax, Valium, Ativan. There's tons of different types of benzos that are used to treat anxiety for the most part, but they're highly addictive. And on top of that, speaking of alcohol, when it comes to withdrawal symptoms, if you're withdrawing on opioids, can that kill you? If you're a heroin addict or you're on fentanyl and then all of a sudden you go into withdrawals, can that kill you? No. Nope. You don't die from opioid withdrawals. You feel like crap, right? So what do opioids do? Opioids cause constipation. Opioids kill pain, right? Opioids reduce your GI motility altogether. It also prevents production of fluids, right? So like runny nose and stuff like that. If you're going through opioid withdrawals, you get pain, you get diarrhea because all of a sudden you have gastric motility goes back to normal. You also get cold and flu-like symptoms. You start like runny nose and all that stuff.
Like hot flashes or like
Everything. Like
chills.
opioid withdrawals are extremely uncomfortable, and that's why opioid addicts go back to doing the drug. Long-term like heroin addicts, if you talk to a person that's on heroin, they usually don't want to do heroin. They get tired of it because it's like, first of all, the first high is the best high. All the remaining highs that they go into, like they need to keep on upping the dose to get that same effect.
I'm sorry.
You're good, dude. They need to keep on taking the drug at higher doses to get that same effect. And at a certain point, it becomes like they're normal, right? And in order for them to feel normal, they have to take the drug to feel normal. Otherwise, they go into withdrawals. So that's the sucky thing about opioids. It's not going to kill you, though. If you want to read a good firsthand description of this, Dr. Carl Hart, I think is his name, Dr. Hart. He's a researcher in Columbia University. He's an actual heroin user, but he's a professor and a researcher on drugs. And he wrote a book called Drug Use for Adults. And in it, he tried to become dependent on opioids just to go through the experiment of going through the withdrawal symptoms. And in the book, he describes in agonizing detail how painful it was, like stomach pain, just muscle pain, everything. It was super uncomfortable. But guess what? He was fine the next day. He just got over it. Now, with that being said, what about alcohol and benzodiazepines? Can those withdrawals kill you? Yes, those will kill you because they throw you into a state of autonomic instability. So how do these work? They work on GABA receptors, right? What does GABA do? It's a neuroinhibitory neurotransmitter, right? So it inhibits. Yes, yeah. So if a person is going through alcohol withdrawals, you want to give them Ativan in a hospital, right? They go, hospitals have usually an Ativan protocol. So they'll give alcoholic patients Ativan so they can slowly wean off the drug, right? Because otherwise, when you get addicted to a drug, what's one of the hallmarks of addiction? You have to use it more and more and more at higher and higher doses to achieve a similar effect. Why?
Because you overcome this.
Where do these neurotransmitters bind to? Receptors. What do your cells do when you have a lot of the thing floating around in your body and you don't need as many receptor binding sites? They downregulate the receptors. If you downregulate GABA receptors and you don't have the drug to stimulate what's left of those receptors, now you're going to get excessive stimulation, right? So you go into autonomic instability. So you become too excited. Alcohol withdrawal symptoms, one of them, seizures. So you can get deadly seizures that are called delirium tremens, right? And so alcoholics and people who are hooked on benzos can also die from those withdrawals. So that's why you want to go on an Ativan protocol. So you slowly taper down the drugs so that eventually your cells can start to upregulate those receptor sites. Make sense? Other drugs that are important, valproic acid. So that's used for seizures, right? Operates on a similar type of pathway, GABA. Gabapentin, it's in the name, right? There's also pregabalin, which is another one. That's a drug that's used to treat neuropathic pain, right? Patients with diabetic neuropathy, they're often prescribed gabapentin, pregabalin. They can help with pain. Propofol, we already talked about that one. What's another name for that? Milk of amnesia. Yeah, exactly. So propofol works on GABA receptors. So basically it stops the brain from working. And then baclofen, that's a drug that can also be used to treat muscle spasticity. 1/3 of all the synapses in the brain are GABAergic. That's a lot, right? Remember I said glutamate, super important. GABA is also really, really, really important in the brain. You find it also in like local circuit, like interneurons, right, between neurons. Here's the synthesis pathway. Again, I'm not going to test you on the synthesis pathway of these drugs, but look at the ultimate precursor, glucose. So glucose is going to be the precursor. Eventually you get the formation of glutamate. Again, that's the neuroexcitatory neurotransmitter. Then you finally get GABA production. And you need a couple different types of co-transporters and things like that to allow for this production to take place. So again, I'm not going to test you on the biochemistry behind all this, but this is the synthesis pathway, right? So you have GABA production from glucose to glutamate. Eventually you get production of GABA. Then it gets released in the synaptic cleft. Then you don't want it to linger for too long, right? So you get funneling back into, again, a supporting glial cell, or it can go back into the presynaptic terminal. So on that note, I think we'll continue next time on this, talking about GABA some more, and then next time we're also going to start talking about some other compounds as well. Serotonergic compounds, also dopaminergic compounds like cocaine and things like that. So that's going to be a particularly fun lecture. So, all right, guys. Have a wonderful day. Enjoy your weekend. And don't get hooked on benzos.